Hi everyone, my name is Maria Manley, and today's topic I'm going to be discussing is polyproteins promote producing polybrium. And basically, to get jump started on this topic, it's basically engineering other organisms to fix nitrogen by combining the required enzymes components into giant proteins that then get cut into the regular size subunit components. Things such as asymmetrical cell division and more. So basically, within this polyproteins promote producing polybrium, scientists have discovered a theory that they developed that could help the different enzymes be produced all at the same levels, so they all have a fair share and could work together, because you can't just have certain enzymes doing more than the other, or certain ones working at a faster pace or another one working at a slower pace. So throughout this topic, scientists have used different methods to study, such as NIF genes, to study the different levels of proteins to see which ones are compatible together, that can work together, because you can't just grab any enzymes and expect it to work with a different one. Like, they all serve a purpose and they all have to be in sync to work together. It's all in one system. So throughout this topic, leading to the four different genes, which are NIF genes. The alphabet of that is H, D, K, and J, which are the three main structural components that the protein needs for the accessory. And there are other ones that are listed as such as E, N, B, Y, and then they use U, S, N, V, and E, and M. But the ones that I just listed right now are not the common ones. The common, or the main four ones, are H, D, K, and J. So moving on, each enzyme has a clump that is the same size that gets turned into protein by the cell separated by a site. So basically, each clump has its own purpose, just like any enzyme I stated before. It can work with any other enzyme. So going further into details, virus is the tobacco in each virus and proteinase enzyme that cuts the proteins at particular sequence. And the viruses uses this to produce its proteins at the right particular location. They tested it out in E. coli, which we then realized like Hg and K inhibited their activity that worked well together. Just like I stated before, you cannot have certain enzymes work with other ones because it's not going to work. Like, you have to make sure they're all in sync and they all can work together. So within them testing it through E. coli, they realized that the first three clumps work together effectively, Hg and K, but not the other ones as I listed, which are like things such as ESB, Y, and so on. And we learned USV messed up the activity cycle. So Hg and K worked sufficiently, but USV messed it up, the activity. So like I said before, the scientists basically created a theory to try to see which enzymes work better together, and they realized when they tested it throughout the E. coli that the USV did not work. So moving on, bacteria. Bacteria, basically on growing plates, when they tested it, they realized that they basically tested it to see if E. coli can fix nitrogen and grow with nitrogen gas as the only source of nitrogen. And it did work, but that's with bacteria growing on plates. So basically, that's what they discovered, like running different tests, running through E. coli and the different letters, they see which ones work better. So then, putting a nitrogen fixing system on any organism that didn't have it before works more effectively and sufficiently when they combine small single genes into large polygenes that make large polyproteins that get chopped into individual components after translation. So after all the tests of scientists from different theories, they had to see which one makes it better, or which ones are in sync together. So basically, when they run the different tests, they couldn't just put any enzymes together. They had to test it with different viruses and things and E. coli to see which ones work sufficiently together. And they realized throughout the different tests that not all of them can do what others can. So going on to the nitrogen fixation, it's a unique process to microbes in the human engineering. It's basically the human process Haber-Bosch that takes nitrogen gas and converts it into NH3 ammonia, uses more energy than the biological process that we learned throughout this lesson. And microbial process is pretty energetic intensively on a microbe scale and pretty tricky. It's pretty tricky because it requires the different components in the right conditions, basically. So then in the Haber-Bosch, we learn throughout the Haber-Bosch process is greatly expanded due to the agricultural and the capacity and made, if possible, to feed our world. So basically the Haber-Bosch process is something that the scientists developed to make sure everybody in the world is getting fed in different things, because as we know, resources are very scarce, so they just can't use anything and everything and expect it to last the whole population throughout the world. Okay, so and it uses a lot of energy and high temperature and pressures, and is using up a lot of resources that we are acquiring from the ground, like I stated before. So basically the scientists started to create different plants that can fix their own nitrogen, or see if different plants can fix their own nitrogen. So the engineering process is pretty tricky as well, just as the microbial process. And the engineering process is basically requiring reworking the plant's immune system. Maybe engineering plants producing their own nitrogen can be easier, as we learned throughout. Okay, and scientists have also developed a system of producing these enzymes in plants with giant proteins that can get cut into smaller pieces of function. So basically, when they grabbed the enzyme, they couldn't just accept the enzyme full, like its full amount. They had to cut it into smaller pieces to see which ones would go and sync together, that would work together and produce the best results. So basically the polyproteins promote producing Pabulum. This was all different tests through E. coli, through asymmetrical division, to see which enzymes work together sufficiently. But yes, the scientists ran different tests, and they basically saw which one was the better deal, which produces the best results, and more. And we talked about the viruses throughout the tobacco and the proteases and the different things with that. So basically, that's what this theory was all about, to see to see if all the enzymes could produce the same levels and have a fair share at everything they do. So not one enzyme can be better than the next, or move at a faster rate than the next, or just do a better job than the next enzyme. This topic was discussing the scientists creating different ways all the enzymes can come together and do one job and produce the best results. But they had to run different tests to see which ones could go and sync together. Like I stated before, they couldn't just throw any enzyme together and see which ones work the best. So yes, all these things I tested did not show anything going on in the plants. It was just in bacteria, so the viruses and the tobacco. Yes, so basically that was it. Basically they wanted to see if it could work in eukaryotic things such as maybe yeast and other eukaryotes, and address other challenges that was involved, such as the scarce of resources such as oxygen, because there's not a lot of oxygen in the world to go around to experiment for each enzyme, And basically, the advance of genetic engineering is made easier by discovering we make out in nature.